Yes, same kind of guy. In this video, we're going to talk about the DNA cross-linking and neoplastics. But specifically, we're going to talk about the nitrogen mustards. Here is the layout of what you are going to discuss. Guys, we have a lot of mechanisms, so please don't forget to subscribe, like the video, and share. Let's go. There are different approaches to cancer treatment. One, there is surgery. Two, we have the radiation therapy. Three, we have the biological response modifiers. These ones, they boost the immune system. And also we have the anti-neoplastic chemotherapy. A combination of two or more of these can be used at the same time, depending on the severity of cancer and the type of cancer and many other factors. These two, they are mainly used for the localized cancer. And these ones, mainly, they are used in the disseminated cancer. Or the systemic. So depending on the severity of the cancer, type of cancer, and many other factors, the combination can be used. These ones can be classified in different ways. One, you can have the structural classification, or according to the functional group. You can have according to the mode of action or the mechanism of action. You can classify them according to the stage of the cell cycle which they affect, and many other ways to classify them. But according to the DNA cross-linking and neoplastics, we are under mechanism of action. Here we have the DNA cross-linking and neoplastics. Also, these ones are many, but among those, we have the nitrogen mustards. What do we need to know about the nitrogen mustards? Briefly. One, they are highly reactive. They have highly reactive electrophilic structure. Since they are electrophiles, meaning that whenever they encounter the nucleophiles, we shall have the formation of bonds. Bond formation with the nucleophiles. For example, in case of the treatment of cancer, we need the bond formation with the guanine. When they form bonds with this guanine, we shall have the alkylation of the DNA, the nitrogen at position 7 of the guanine. There are many other nucleophiles, for example, the adenine. They are not cell specific. They are not cell cycle specific. Meaning that at any stage of the cell cycle, they can affect the cells, but mostly they affect the the G1 and the late G1 phase and also the S phase of the cell cycle. But also in other stages, they can affect the cell. Also, these drugs, they have ability to cause carcinogenesis. Though they treat cancer, they can cause cancer. They have ability to cause carcinogenesis and mutagenesis. Because mutation can lead to cancer. These drugs, some of them are very scant. Some are very scant. We are going to see them. Meaning that in case they come into contact with the skin, they can lead to blistering. And also we can have necrosis. So for these ones, meaning that they are always intravenous, they are IV. They shouldn't come to the skin or extravasation of the blood vessels. Also, sometimes they can alkylate or they can form bonds with the guanine of the same gene A or two guanines from different gene A strands. So they are bifunction, meaning that they alkylate two gene A molecules on the same drug, but mostly on different gene A. It can be the guanine of the same gene A, two guanines from the same gene A, or guanines on the different gene A. Hence, they are cross-linkers. These are the few things we need to know about the nitrogen mustards. Let's go to the genome structure and talk about the mechanism of action. Yeah, it's same color guy. For the genome structure, these nitrogen mustards, they have this structure. This is the genome structure of the nitrogen mustards. We shall see some deviations, but the genome structure is this one. This is the alpha carbon, and this is the beta carbon. The changes, we shall see the changes only at the R group, but these other structures, the ethyl chloride, they are the same. For example, if it's a microethamine, it will be like this. The R group will be the methyl group. This is microethamine. When it comes to the merfuran, it will be the N. The R group now will be this. This will be the R group. That is merfuran. So these groups remain, but the R group is the only one which changes. When you talk about the bendamustine, we shall have this. This is bendamustine. This is melphalan. When you talk about the cyclophosphamide, it will be like this. This is the cyclophosphamide. When you talk about the ifosfamide, it will be, now these are the changes I'm talking about. We shall have, instead of the alkyl group, instead of the haloalkyl group, we shall have, one will be the hydrogen. Then this one will be the haloalkyl group. Then we have this. One of the haloalkyl group now will come to this nitrogen. This is the ifosfamide. So these are some of the examples of the nitrogen mustards. We are seeing that it's only the R group which changes, apart from this ifosfamide, where we have the one of the nitrogen mustard arm, one of the mustard arm goes to the N of the ring instead of this N. Let's see the mechanism of action.